We're very pleased to have Dr. Klaus Kaestner joining us from the Human Pancreas Analysis Program, HPAP, which is part of the Human Islet Research Network, HERN, and he's coming to us from UPenn. HPAP is responsible for, sorry, I'm just letting people in, is responsible for performing deep phenotype of the human endocrine pancreas to better understand the cellular and molecular events that precede and lead to beta cell loss and or dysfunction in type 1 diabetes, T1D, and type 2 diabetes, type T2D. The the NIDDK supports HPAP through multiple NIH grants. And a little bit about Dr. Klaus Kaestner. He's the professor of genetics at UPenn School of Medicine and a member of Penn's Institute for Diabetes, Obesity, and Metabolism. He received his PhD from Johns Hopkins University School of Medicine and completed a research fellowship at the German Cancer Research Center in Heidelberg. His work is focused on understanding the molecular mechanisms of islet cell function and glucose homeostasis. His laboratory pioneered the development of functional genomics tools applicable to the study of the endocrine pancreas. Currently, he is the co-director of the NIDDK-funded Human Pancreas Analysis Program, HPAP, which we're going to talk about today. And this focuses on improved understanding of both T1 and T2 diabetes. They are using deep profiling of pancreatic islets and other relevant tissues from deceased organ donors. So I'm sure that everyone feels very thankful to those families who have donated in that capacity. He's served on multiple review panels for JDRF and the NIDDK. He serves as co-editor-in-chief of Cellular and Molecular Gastroenterology and Hepatology, CMGH, and on the editorial boards of Molecular Metabolism, and is associate editor of the Journal of Clinical Investigation. In 2017, he received the Roy O. Green Award for Outstanding Research in the from the Endocrine Society. Thank you very much for joining us, and we really look forward to hearing about your role at HPAP, the progress of HPAP and the future for HPAP. So yes, thanks for joining us.
Thank you so much, Monica, and thanks everybody for joining. I will try to share my screen to, oops, you disabled participant screen sharing.
Oh, try again.
Sure. Aha, there we go. All right.
Perfect.
Yes, so thanks again for being here. I'll tell you a little bit about the Human Pancreas Analysis Program and how we're trying to enable new insights into type 1 diabetes. So when we think about the pathogenesis of type 1 diabetes, this is sort of the Eisenbart model, which was developed some 30 years ago or so, which tries to summarize the events that actually happen. So if you think of a pancreatic islet shown on the left here, you have about equal numbers of beta cells shown in green that make insulin, and alpha cells shown in blue that make glucagon, and a few delta cells mixed in between, and a few minor cell types as well. Over time, beta cells being attacked by the immune system and eventually end up with an islet which is the alpha and delta cell only, and no more beta cells are left, or very few beta cells are left. So that's what happened on the tissue level. And what happens in the patient is that we envision that functional beta cell mass slowly declines over time until at the end in stage 3, the beta cells are left are not sufficient to maintain homeostasis, and therefore you end up with symptomatic type 1 diabetes. Recently, the disease has been subdivided into 3 stages. Stage 1 would be individuals that have 2 autoantibodies but are still normal glycemic. Stage 2 would be those where dysglycemia can be discerned already, or they're not yet openly diabetic. And of course, then stage 3 is the symptomatic phase of disease. We know that autoimmune process proceeds during all these 3 stages. And what HPAP is trying to figure out really is what's happening before here. What we do know is that there's variable genetic and environmental risk for type 1 diabetes. The genetic risk we understand the best. We know that there's certain alleles of the HLA loci that are specifically associated with a higher risk of type 1 diabetes. Some can give a newborn child a 12-fold higher risk than the general population of getting type 1 diabetes. Nevertheless, of course, type 1 diabetes is not Mendelian. In other words, your genotype is not deterministic of your phenotype. There's many people with risk alleles for the HLA that never develop type 1 diabetes, and conversely, there's many people with type 1 diabetes who do not have the high risk alleles. So we really don't understand completely what the predisposing variables are. Then an as yet rather undefined triggering event is inferred that then initiates the autoimmune response. Many different factors have been invoked here, various viruses have been proposed, but to the general understanding in the field is that as of today, we do not know precisely what the triggering event is. Otherwise, we would be able to interfere with it, but we cannot. So while a lot of knowledge has been obtained from mouse models of type 1 diabetes, in particular the NOD mouse, and a lot of current work is focusing on studying live patients and their relatives. For instance, the very large TEDdy study that collects blood samples from relatives of type 1 diabetics to monitor at least the events that change in the bloodstream. Very little work has been focusing on actually studying the earliest events that happen in the pancreas itself. The reason for this is that it's actually impossible to safely biopsy the pancreas, hence very little tissue can be obtained. So what these sort of formed the rationale for HPAP T1D, which was initiated in 2016 through an effort of the NIDDK. And these are some of the rationales that were brought up by the group. First, over the last 10 years, a lot of new experimental technologies have been developed that allow the studying of the immune cells, endocrine system, and the pancreatic compartment. And these require really specialized organ procurement and processing. There are many experiments that we do in HPAP cannot be done on historic FFPE fixed tissues. It's simply not possible. So we needed to have a new program for this. Secondly, it was felt that it's critical to study both isolated islets, the pancreatic tissue, and the associated lymphoid organs of the relevant patients in great detail and correlate that with clinical data. Third, turns out that in the United States, the number of new onset type 1 diabetes cases that become available from organ donations is rather small, maybe less than 10, and therefore this requires a centralized organ procurement and therefore also tissue analysis program. And finally, we thought from the very beginning that rapid data sharing is needed to advance the knowledge about T1D pathogenesis, and I'll go into the detail of how HPAP in a very unique fashion deals with data sharing. Okay. So Monica already mentioned this, but I'll repeat this briefly. The mission of HPAP T1D is to perform deep phenotyping of the human pancreas and its interaction with the immune system to better understand the cellular molecular events that precede and then lead to type 1 diabetes. The goal of HPAP is to accumulate, analyze, and distribute high-value data set to the diabetes research community. Of note, I'm only representative here of a group of the leadership team, which is Mark Atkinson in Florida, Ali Naji at Penn, Al Powers at Vanderbilt, and myself. And these are the institutions and the grants that support this work. And of course, without the donors and their families, we couldn't do anything. So how does HPAP proceed? First, organ donors of interest identified through the NPART network and relate to the HPAP team. This, of course, involves the screening for autoantibodies through organ procurement organizations all throughout the United States, which has been organized very well through the NPART team in Florida. If a suitable organ donor is identified, the pancreas, but also the immune tissues are all shipped to Penn for further processing. Of course, out of this, we get tissues as well as islet cells. And briefly, to summarize, the detail of how we do this, just about the pancreas, for instance, we actually collect tissue sections from the head, body, and tail of the organ, and these are processed in multiple ways to allow analysis using multiple modalities. In addition, of course, pancreatic islets are isolated, and these are analyzed for function, for metabolic changes, and actually even for immune biology. And of course, also a lot of molecular phenotyping is being done. The actual phenotyping is done in laboratories at Vanderbilt by Marcela Brisova and Al Powers, as well as at Penn by Doris Stoffers and myself and all the immunologists. And we perform tissue analysis, very extensive molecular profiling with the most advanced techniques available, islet physiology, several methodologies for multiplex imaging and immune studies. And importantly, all these data we obtain are deposited into an integrated database called PancDB, and these data are made available to the world immediately. All that's required is that a researcher register the name and information and agree to acknowledge that the data were obtained via the HPAP program in any publication that results from the use of these data. And that's it. So we're not waiting for publication of the data, but rather we make them available as they're being produced.
Can I just jump in here? How much data is currently sitting in that PancDB?
Literally gigabytes of data. Many thousands of different files are in there. And they have different modalities, from images of the tissue, from immunostaining, data from single-cell RNA-seq of 250,000 cells from the pancreas, 600,000 immune cells, all kinds of different modalities. All those data are within PancDB.
And the TCR data, for instance, is it standardized, and I guess sort of harmonized with other data profiles?
I believe so, although this is the work of the immunologist that I'm less familiar with. So, but I do hope so, yes.
Okay, cool.
Good point. So where are we this far? As of November, we've been able to obtain 21 organ donors with type 1 diabetes, and these are in quite a broad age range from 2 years to 37. And we also have individuals with no history of diabetes, about 50, and again, very importantly, within the same age distribution. Now, of course, people without diabetes are actually divided into two types. There are those which have no autoantibodies, and then about half of them are autoantibody positive, but still normal glycemic. These, of course, are very interesting individuals, as we hope to be able to determine the very earliest events in the autoimmune attack within the tissue itself using these tissue samples. The distribution via sex is also quite nice, about half and half male and female. The only different distinction here is the autoantibody positive that's skewed towards the male donors. And also, if you look at ethnicity, we've been able to roughly represent the distribution of ethnicity within the United States with a fair representation of Hispanics and African Americans as well. So what do we actually do? Of course, immunobiology is at the center of HPAP T1D. And the goal here is to develop an immune atlas of lymphocyte populations, their phenotype, their clonal interactions, and antigenic response, epigenetic transcriptional signatures. Of course, blood is collected, but blood, of course, is available to many programs like TEDdy, but what they don't have in TEDdy is, of course, lymphocytes from the spleen and the lymph nodes surrounding the pancreas. And here, it's quite notable how a transplant surgeon, Dr. Nagy, is able to collect pancreatic lymph nodes from the tail, body, and head of the pancreas separately and make them available for analysis. In addition, we even obtain lymphocytes that migrate out of the islets during culture, so you can actually look at islet-resident immune cells as well. And down here, some of the leaders in the immunology analysis program. So phenotypes are very complex, determining or deriving an immune atlas of innate cells, B cells, and CD8-4 and CD8 T cells, as well as doing functional studies. I'm just going to give you a couple of brief examples of what has been found, and to actually look at sort of what we call innovative immunobiology of type 1 diabetes. Because lymphocytes are collected from the supernatant of the islet culture, these were then subjected to 24-color cytometry to identify both memory and tissue-resident CD8 T cells. And if you do this and you compare the healthy controls to type 1 diabetic, it's very obvious that the T cells within the pancreas of type 1 diabetics are angry, in that they're activated as seen by Ki67, a marker of proliferation, as well as the expression of coenzyme B and perforin, which again are markers of activated T cells. So it's really a very, very dramatic difference. And of course, it makes sense in the disease, but this was a lovely way to document it. I should also note that my lab, who does the imaging mass cytometry, was confirmed this on the tissue section using a different methodology as well. Now I want to tell you about two unexpected findings that came out of HPAP. And I think this, to me, documents how useful it has been to establish this program and to analyze human tissue sections. So we go back to what happens in type 1 diabetes to the islets. It's very clear, of course, we have the loss of beta cells, and that's what the whole world has been focusing on. However, we actually, through HPAP, were able to identify a new defect and surprising defect in pancreatic alpha cells. So we started, as I said, the analysis always includes hormone secretion studies. This is in GAD autoantibody positive individuals, so that we considered stage 0 because they don't have 2 autoantibodies, only 1. And you can see if you look at insulin secretion, comparing the controls in black to the autoantibody positive in red, the curves are identical. And what you see here is you get nice stimulation of insulin secretion with 16 millimolar glucose. G stands for glucose here, so nice stimulation. And if you add an agent that increases cyclic AMP level called IBMX, you increase insulin secretion even further. So that was known before, and as expected, the single autoantibody positive beta cells function perfectly well. You can, if you quantify this to the area which is the glucose stimulation here, again, there's no difference. If you look at the IBMX stimulation on the right here, again, there's no difference. So the surprise came when we looked at the glucagon secretion, which is not often done, but we done by our HPAP program here. And where glucagon secretion works, of course, the glucagon secretion is suppressed by glucose. So in the black curve are the controls here. You have stimulated glucagon secretion, then you dial in glucose, glucagon secretion is decreased, you put in more glucose, you secrete even further. Remarkably, if you do this in islets from autoantibody positive individuals, the glucose suppression is not working, or not working well at all. And this is the quantification at 3 millimolar glucose, statistically significant difference between the two groups. And the same is true at 60 millimolar glucose. Again, a dramatic difference in the suppression of glucagon secretion. So this is new, novel biology that was not known before. The nice thing about HPAP is that we, of course, obtain data on the same organ donors in multiple modalities. And what we had was single-cell RNA-seq data from all these islets. So that allowed us to select only the alpha cells from the single-cell RNA-seq data and look at gene expression in those alpha cells. And what we found is quite remarkable in that on the left here, you see something called a gene set enrichment analysis, where we can look at all the genes that are involved in glycolysis and gluconeogenesis. You can see on the top, these lines here are individual genes in these pathways, and you can see that a whole bunch of these are down-regulated in the GAD-A single autoantibody positive individuals compared to the controls. And if you look at some of these genes individually, you can see very famous genes here. Glucokinase, it's well, for instance, is in here. And the red dots here are always the GAD autoantibody positives, the blue controls. And in every single case, you can see higher expression in controls and lower in the autoantibody positive. And this is actually very satisfying because you need glucose metabolism through glycolysis and oxidative phosphorylation to turn off glucagon secretion in alpha cells. And therefore, if this whole set of enzymes is expressed at lower levels, glucose will not be as effective in suppressing glucagon secretion. Again, a completely novel defect in alpha cells seen in already in single autoantibody positive individuals. If you, and this actually fits nicely here in the middle of the GAD autoantibody positive individuals to what was described by Marcela Bissova and Al Powers before on alpha cells in type 1 diabetic patients. So these with frank diabetes, which also had found altered alpha cell gene expression and loss of glucose suppression. We now document that this actually starts to occur in single autoantibody positive individuals. Now, this raises an interesting possibility, and that is, could it be that in the earliest stages, stage 0, if you wish, of the autoimmune disease, that defects in the alpha cells contribute to the triggering event in the autoimmune process. This has to be determined in the future, but I think it's interesting to note that many of the proteins that are present in pancreatic beta cells are, of course, also present in alpha cells, and among them GAT65, which is the autoantigen to which these autoantibodies are reactive to. So that was surprise number 1. And then briefly, surprise number 2, and that has to do with how our multimodal analysis of single cell data was able to tell us something about novel states in type 1 diabetes. So here, same principle, we're comparing controls, autoantibody positive but normal glycemic individual type 1 diabetics. In this case, we did single cell RNA-seq at that time of 80,000 cells, 1 million cells, which we quantified by imaging mass cytometry, where we can look at 40 different proteins at the same time, and 7 million cells by flow cytography, again with 40 proteins, which we developed for pancreatic islets. And we can then again integrate all these data together. And there's 2 small interesting findings here. The first has to do with the GAD autoantibody positive individuals, all normal glycemic. It turns out, of course, the levels of GAD autoantibodies vary in the blood of these people, right? So you can then plot increasing GAD levels. You can actually correlate that with a specific gene signature in one of the beta subtypes called the beta 1 cell type. You can see that these genes, about 1,500 of these beta cell expressed genes increase as high and correlate very well with the level of GAD. How this happens and what this means, we don't know yet, but it's certainly the first time that this has been described. Secondly, and this was even more puzzling, data came from when we actually looked at the pancreatic ducts, which we normally ignore because all they do is they take digestive enzymes produced by the exocrine pancreas and conduct it to the duodenum, right? And you can tell pancreatic duct cells by staining for cytokeratin, shown in red here. What we noticed was that, in fact, many of these cells are also positive for HLA-DR, which is one of the class 2 HLA proteins. You can see many of these cells are now also green, so double positive. And this suggested then something very strange, how ductal epithelial cells might have a role in antigen presentation. So because we had single cell RNA-seq data, we can actually dig into this a bit farther. Oh, sorry, before I get to that. These cells are present in every individual. A small subset of the cytokeratin positive ductal cells also express HLA-DR. But when you look at the different donor groups, you can see that the vast majority of these occur in type 1 diabetic individuals. If you plot this by group, again, highly statistically significant increase in these strange cells in the type 1 diabetic pancreas. And as I said, when we looked at the gene expression of these particular cells, they're subsetted for being cytokeratin positive and expressing HLA-DR, we found that they actually expressed a gene set that had been described previously for dendritic cells, specifically those dendritic cells which adopt a tolerogenic phenotype, meaning these dendritic cells actually suppress the T cell response instead of stimulating it. And in fact, this gene set is highly enriched in these unusual ductal epithelial cells in the type 1 diabetic pancreas. So the way we interpret this is as follows. Exocrine ductal cells might acquire a signature of tolerogenic dendritic cells in an apparent, although of course ultimately unsuccessful attempt at immune suppression in the type 1 diabetic pancreas. And again, this is a finding that's novel out of HPAP. So lastly, then I want to tell you about what, of course, the very important goal of HPAP is to distribute high-value data set to the diabetes research community. And how do we do that? Well, the one, of course, is obvious through our publications. And it turns out since 2018, HPAP contributed to 21 papers in really high-profile journals. But notably, out of these, only 11 are peer-reviewed publications published by our own team. 7 actually were papers coming from outside users who simply took the data, analyzed themselves, and published their own paper, most of the time without us even knowing about it. Okay, So how do you get access to the data? This is done by our database called PancDB. This was upgraded just in May, so that now not only can you download donor information on all the experimental data, but you can even perform interactive analysis on the data sets, in particular gene expression data, directly on this database. Sorry, excuse me. And just in the 6 months period here, you see that we had 154 individual users who downloaded over 35,000 files from HPAP. And again, you don't have to ask permission from anybody. All you have to do is go to the website, register, and say you agree to cite HPAP in your publication. And that's all that's needed to gain access to the data. And here are actually some of these papers, for instance, one from Dezia Eiserick in iScience recently looking at the transcriptomes of alpha and beta cells comparing type 1 to type 2 diabetes. Great study. We weren't aware of this, but good use of the data. Another one of targeting self-antigens target T cells, again, making use of the data, and so forth and so on. So we're very happy that the community at large is actually making use of the data that HPAP is producing. So to summarize then, we are very grateful to the donors and their families. Without that, there wouldn't be any HPAP. Leadership team I already introduced, and these are the principal investigators that conduct various aspects of the work that HPAP has done. So I thank you for your attention. I'll be happy to take any questions.
Thanks, thanks very much, Klaus. I'd like to open it up to questions. If anyone has a question, you can drop it in the chat, or you can actually unmute yourself or ask to unmute yourself, and we're happy to entertain these questions. Silence. I don't see any hands up yet. Okay, so yes, great presentation, great resource. Thanks a million. That's what someone has said here.
Much appreciated.
I wanted to just kind of press a little bit about, okay, so now this shift of maybe for some individuals or this potential endotype of the alpha cell goes offline first, right? Mm-hmm. Or just dysfunctional. So in those cases, when you looked at the histology of these, individuals later, can you see, the cellular defects in the alpha cells, or do you see a disappearance of those alpha cells if they've, like you see with the disappearance of the beta cells, or is it just,
Right. Yeah, an interesting question. So, of course, as is the nature of human subject research with deceased organ donors, we have exactly one time point, right? So, and this is all people who are still normal glycemic. And we did, in fact, do very careful islet morphometry. We stained multiple markers for alpha cells and beta cells, and alpha cell and beta cell mass are completely normal in these individuals. So the alpha cells are still there. They're just not functioning normally. Yes, thank you.
And then, what are your thoughts around just sort of, this is a speculative, of course, but, developing a therapy for those that do have this alpha cell dysfunction? I mean, because primarily, the big targets up until, recently have been, resurrecting the beta cell or dampening the immune system. But if you want to talk about the alpha cell now as being center stage or maybe primary for some individuals, how might you address that?
Very, very, very good question. And I think the answers differ a little bit when you think about type 1 diabetes and type 2 diabetes. So I think for type 1 diabetes, first of all, be curious to figure out why the alpha cells misfunction. I do not know that, and we have no idea at the present time. Is that, in fact, a consequence to the initiating immune attack, or is it one of the cause of this? So we do not know that one way or the other. And the next thing would be that if it were to contribute to the immune attack, would have to determine what are the antigens that are presented both by alpha cells and beta cells that might be initiate this process, might be involved in this. So that's sort of, I think, way more of a research side, research project, which might take many years. For type 2 diabetes, I think there we're a lot closer in that many people have argued for 30 years that type 2 diabetes is actually bihormonal disease, meaning you don't have enough insulin secretion, but you also have too much glucagon secretion. And there are, in fact, targets in the glycolytic pathway that can address both of them at the same time, meaning there would be, you could decrease glucagon secretion and increase insulin secretion at the same time. So that, I think, is a little bit closer to becoming reality.
Okay. And I also just wanted to give a shout out here to the collaborative process. You know, you've got Decio Zurich at the ULB, so across the pond there, right? And so it's a European reach there. And I think, you know, the more collaboration between great minds wherever they are is important. So that's a very nice feature of this program that you've got going. Also, you know, finally, we're at the end of our time here, but I would like to say that, you know, what is your what is your expectation for the, you know, evolution in the coming year or two of HPAP?
Yeah, so I think that there's two parts. One is methodologically, and one is scientifically, I guess. Methodologically, the good news is that in the molecular field, there's constantly new developments. And one thing that we'll be adopting starting in March is a process or methodology called spatial transcriptomics. Yeah. There's now methods available that can actually do this at the subcellular level, and with looking at more than 1,000 transcripts at the same time. So we're collaborating with a company to make a panel that's specific to the genes that are relevant to the human pancreas. So that should be able to do that in March. And the nice thing there is we can do this going all the way back with archival samples, back to HPAP case number 1, and then fill in the data on this. So that's the way that goes. The other thing that we are quite interesting is to expand our numbers, especially as it comes to the different ethnicities, right? So we want to know, are there differences in the, you know, immune tissue interaction in the pancreas depending on your ancestry, basically. And as we're recruiting more and more of people of African ancestry as well as Hispanics, we might be able to actually get to that point in the future.
Okay. Here's from George Harb. Thank you, Klaus. Great to see mechanism uncovered behind the positive correlation between the number of ductal cells implanted and long-term metabolic success in allo-islet transplants. Yeah, so for the people that are interested in islet transplantation, the vertexes, et cetera, of the world, right? This is important and relevant information. Okay, well, thank you so much. I really appreciate your time with us. It was informative and it was inclusive, and it looks like, you know, there's a lot of value to be had for those in the scientific community to do some deep dives into HPAP. And I
thank you so very much.
Thank you again. Have a great rest of your day. Bye-bye. Okay.